Okay, everyone, many thanks for your patience. We've had a few technical issues, but it's my great pleasure to introduce Glen Fox, who is been appointed Professor of Malting and Brewing Science, the Anheuser-Busch Endowed Professor of Malting and Brewing Science at UC Davis. And he's here to give us a talk on malt chemistry, the importance of protein content and protein composition.
Well, good afternoon for everyone there. It's about 9:10 in the morning, California time. I'm delighted to be with you and giving this presentation. I know I have attended a few of the previous webinars and really enjoy the content and how it's all engaging with everyone from academia through to industry. So hopefully if everything works, I'm just going to present some work from a previous couple of students, both here at University of California, Marnie, who's now graduated with her PhD, my colleague Christine Dykenbrock, who's a plant breeder here in UC Davis in the Plant Sciences Department, an undergrad who did a lot of the work. As you know, the undergrads do the work, the rest of us just take the glory, and a previous student at University of Queensland, where I'm from originally. As you can probably tell, I do not have an American accent, and all hopeful that I will never have one, because I'm contemplating retirement, and that's possibly happening sooner rather than later, given the current political environment. Moving on from that, let's not talk about that at all. I'll cover the motivation for this particular topic, and as both sort of from an academic perspective as well as an industry perspective. We'll go through grain composition, which I'm sure everyone is very, very familiar with. But we'll probably dive into some aspects of grain composition that a lot of people won't be terribly familiar with, I don't think. Or maybe I'm just a little naive and thinking people don't quite understand some protein composition, and maybe everyone's really full bottle on all of this. I was giving a presentation last week to our Master Brewers class, and these are brewers from breweries around the world, and a lot of them didn't really have any background in understanding of protein chemistry per se. So maybe it is a little bit on. And I'll talk a little bit about the relationship of protein, and we talk about protein content as well as protein composition to the other grain constituents, and possibly a new method to measure storage protein content at the same time as measuring total protein content, which may give maltsters at least some guidance on the maltability of some of their samples. And then, of course, a brief summary. So in terms of the motivation, and this is something I've banged on a long time when I was in Australia, and I've been here in the US 6 years now, and I've been banging the same drum. And there's some movement in some industries about shifting their focus on how they get their malt analysed. And you guys in the UK, I believe the UK methods, or the original Institute of Brewing methods, were the better methods to measure malt extract, because you use that 65-degree infusion mash. That was all harmonised with the EBC about 10 years ago, so that method is sort of in the EBC extract method, sort of way down the list there. But quite honestly, I think, and a number of maltsters agree with me, that this is the better method to actually show the capacity of the malt in the brewery. The EBC method allows other things to happen, and the US is a good example. 90% of breweries here are making IPAs, so they're mashing in around 65 degrees. So the malt COA is probably not telling them a lot, and maybe actually giving them some misguided performance information. So that's the drum I've been banging for quite a while, is actually getting more relevant data that the maltsters can provide to the brewers about what the malt will really do in their brew house. Maybe not what they think the malt might do, but really what the malt will do. But we also know there's a limited number of parameters on the malt COA. And you can talk to maltsters and they go, yeah, we can change the malt COA, but what do we change it to and what do we take off? You can't just keep adding things to the COA realistically. And the other downside is that a lot of the craft brewers I work with here in the USA, they can't even spell malt COA. No, that's a joke, sorry. But they probably don't see it. The head brewer might see it, and that head brewer might make decisions about recipes, but the other people don't see the malt COA, so they're not really sure of the malt quality they're handling. They're just doing the mashing process, etc. So that's one of the motivations. And the other motivation is really about how complex quality is, whether it's grain quality or malt quality, or even hop quality. That's another conversation about hop chemistry. But it's a lot more complex than people think. Often they're thinking, well, we measure protein content. Great, you've measured protein content. But what does that tell you about enzyme potential? What does it tell you about foam potential? What does that tell you about haze potential? They think, well, it's higher, so I might have more enzymes, higher protein content, theoretically more beta-amylase. I might have more foam-positive proteins. But that really doesn't tell me how the mashing might go. Will I get an efficient mashing process? Those sorts of things. And I think we need to really, and this is, and I know you've got students there who've been working on projects to look at the impact of climate and how it's going to impact grain quality, malting efficiency, all those sorts of things. But living in California and coming from Australia, we're used to seeing summers when they're harvesting the grain crops well above 35 degrees C, and I can use the C word with you guys. Here I have to use the F word, which is fine. But even in the US, and I know even in Europe, they're seeing sort of the springtime and summer temperatures where they're harvesting the grain well in excess of 35 degrees C. And the maltsters are telling me that they're concerned that they're just not seeing the quality and the grain quality specs and not giving them enough information about how things will malt because the composition is changing. The protein content might be the same, but the composition is changing. And that's to do with how all of these components have been laid down during grain filling, which is a very complex process. So hopefully we're all very familiar with the barley grain. That's what it looks like. All the components of the grain, the starchy endosperm. For probably the best part of 10 years before I came to the US, we were working on starch quality and looking at the impacts of starch structure, gelatinization, mashing efficiency, production of fermentable sugars, and then fermentation efficiency. And that was all very good. And now we've got to the point where we're seeing maltsters on a regular basis asking for gelatinization temperatures. So I'm actually literally, when I get back to work on Monday, I'll be testing some malts for a maltster for their gelatinization properties. And the concern is we're seeing gelatinization temperatures well above 70 degrees C, which means it really does have an impact on mashing efficiency. But at the same time, if we're seeing these variations in starch quality, then it translates to variation in other grain components, because it's not the starch in isolation that's might be impacted during grain filling. That whole complex metabolic, cascade of pathways are all being altered in some way due to heat or drought stress. Anyway, we'll get to that. So these are the SEM images I love. We see a beautiful image there of our starch granule, and that's on the bottom left. And adjacent to that is where we see the large starch granules, some small starch granules, and then this protein matrix that surrounds those granules. Now, the large granules, they might have a relatively small interaction with the protein itself because they're very large, and the protein might only actually bond to these granules in certain places. But the small granules might be more impacted and could almost be completely embedded in the matrix. And it's difficult to see there, but I have another image, and it's clear there's a lot of granules that are completely bound up in the matrix. So during malting, the amount of modification of that storage protein that has to happen, so those small granules will be potentially more available when the starch enzymes have to do their business. Excuse me, I am recovering from the flu. So bear with me, and I will be taking a lot of coffee during this. So people will be familiar with seeing these sorts of things, protein map as a consequence of a yield map. This is one farm about 6 weeks before harvest, and that was the final yield map of that farm and the protein map. So we harvest that grain, we measure the protein content, and that was 12%. So what does that mean? Well, we know there's significant variation across the field because we already have the yield map. And even though when we put all that grain together, the average protein content of that crop was 12%, clearly there are portions of that field where it's in excess of 14% protein. And some regions of that field or some areas of that field, it's probably as low as 10%, but it's all bulked together. Most of this is driven by environment. As we know, some of it will be driven by fertilizer inputs, those sorts of things. Small portion given by variety. If we think about 12% protein content, and we know we have that inherent variation, well, then we can actually see what the variation might look like by actually testing individual kernels. And I did have an undergraduate who, believe it or not, was very keen to do this. I have done this myself, but not at the scale that she did it. And she counted out a couple of thousand individual kernels from a couple of these fields, 2 different varieties. And we developed a single kernel NIR calibration. So while she had to count out a couple of thousand kernels, she wasn't doing Dumar combustion on a couple of thousand kernels. We built the NIR model. And she was just scanning each of these kernels. And that was the range we saw in those 2 samples. The pinkish colored sample is averaging around 12% from that field. An adjacent field, much lower protein content. But again, we see this huge distribution. Now, this is where some of the inherent variation comes that the maltsters will have to deal with. Even if they're taking a single load from a single farm going into the malt house, we know in the commercial world where you're malting 100,000 tons a year, that's challenging. So you're taking multiple loads, storing those, accumulating enough grain, hopefully within a relatively narrow protein range, and then taking that into the malt house. And all of that is bringing in more and more variation, even though you still might be hitting that 12% protein range or 12% protein target. So we see this inherent range. So they get this inherent range in protein content, then you get inherent range in all these other characters. You get inherent ranges in protein content and protein quality, inherent range in starch content and starch quality, beta-glucan, arabinose, everything is varying in similar relationships here. If we go back, and I do love talking about history, I'll bore my students to death talking about history, but sometimes history is very important to help us understand where we are. So if you've done any protein chemistry at undergrad level or even graduate level, where you might have done electrophoresis, this is probably a bit of a refresher. So Osborne, well over 100 years ago, was classifying proteins based on their solubility. So, and these were the 4 solubility groups we got. Albumins were the water-soluble proteins. Globulins were soluble in water with a little bit of salt. The prolamins were soluble in alcohol. And then the glutellins, the really high molecular weight, were soluble with alcohol. And a reducing agent, something like dithiothiazole or mercaptoethanol, those really stinky sulfur chemicals. So they were the 4 classifications based on water solubility, but sometimes that tells you zip about the functionality of these proteins. So in recent years, we've had these classifications based on functional properties. So fitting in within the albumin and globulin family, we have these metabolic and protective proteins. And over on the right, we have our storage and structural proteins, which is to do with alcohol solubility. But not all alcohol-soluble proteins are storage proteins, and we see these large storage proteins as well in the endosperm. Now, that's all great. It tells us potentially a lot more functionality. Now, even this basic figure here is showing us about 20 different proteins. So suddenly we can sort of imagine going from, we've got a grain protein at 10% protein content. And it's telling us we've already got 20 proteins that we know are involved in coming to that 10% protein value, 10% protein. But what's the proportion of all of those? And this is where we'll get into some other more detailed decomposition of all of this. So yes, that image on the right, top right, is me doing a Keldale way back in the 90s. Yes, I am an old bugger. Been around a while, and doing Keldale was the go-to method then. And Keldale came up with his method at the Carlsberg laboratory in Denmark well over 150 years ago. And anyone who's done a Keldale, old school Keldale, will know it's a very dangerous method. You're basically boiling an organic sample in sulfuric acid, 2 molar sulfuric acid, and those elements on that middle figure, that's basically belt height, so down here at your belt or slightly lower. So this is boiling sulfuric acid. At the top there, after you sort of boil that, you can then take a sample of that, and you can boil that, distill off the ammonia. So in that top blue flask, I'm actually boiling with caustic. So you're either going to burn below the belt or you're going to burn at your face. It was a very dangerous method. Fortunately, the Keldale methods are a lot more safer nowadays, but still rather environmentally unfriendly. Dumas came up with his method about the same time as Keldale, but the technology wasn't there to sort of really give his method any credibility. But thankfully, in around the 1980s, people developed these combustion methods, which is based on Dumas' principle. Now we have these LICO and Elementar and all these other combustion instruments, and this is where we can now actually measure a single kernel of grain. So we can get the protein content of a single kernel of grain, which was very helpful to allow us to understand this inherent kernel to kernel variation. So that was measuring content. Now, if we want to measure composition, again, there'll be a lot of us in the audience that have done these methods before. So you could do one-dimensional electrophoresis, where you might separate proteins of a single solubility group based on molecular size. So that image on the bottom left is a standard electrophoresis gel, where you can see different patterns there, and you can probably clearly see that there's possibly differences between those bands or those lanes on that gel, and possibly differences between the samples. It looks like there's duplicate lanes there per sample. So we can say, yes, there could be differences in those samples based on the electrophoresis result. We can do the same thing with charge. Proteins are charged, so you can separate proteins on their charge. They could be more positively charged or more negatively charged. That would be isoelectric focusing. Then some really clever folks said, well, let's put those two together, and you could do a page separation. So you're separating on size, and then you could put that on an isoelectric focusing gel, and you could separate on charge, and then you start to see these spots. So suddenly we might go from seeing 10 bands on a gel to seeing 50 or 60 spots. And this is where it's starting to say, well, there's more in this than we can see, and certainly a lot more than we'd see if we're looking at a standard electrophoresis gel, even though I know people still do electrophoresis, and I think it's a great technology. So the 2D electrophoresis were what we call the first iteration of proteomics, where we could see all these individual proteins. Now, the image on the sort of bottom right there, that colorful yellow and orange image, is some proteomics we did about 10 years ago, looking at foam positive proteins and where these foam positive proteins are coming through the mashing and brewing process. So now we're going from measuring, maybe getting a result with 10 proteins based on their solubility, to maybe 50 or 60 based on their solubility group initially, but also the molecular size and charge. Now we're seeing literally 200 or 300 proteins. And the proteomics does not worry about solubility. It can basically, with the enzymes we use, digest any proteins of any solubility class, and allows us to see this huge variation in the protein composition. So again, if we look back at the sort of storage protein matrix, in barley we talk about the hordeins and glutellins, although that's usually just encompasses the hordein family, and they're what we call the storage proteins, or the major storage proteins. Of those, the hordeins and glutellins make up about 80%. Now, this is where, again, I think this is one of the key understandings, not from my research, because this isn't new at all. I remember Stuart Swanston from the Scottish Crop Research Institute was doing work with Melina Cano from Spain in the early 2000s, looking at some of this stuff with Triumph mutants. And there's been other people work on this on and off for the last 20 years. Actually, in the 90s, as a PhD student in Australia, Christy Howard was looking at particularly the D-hordeins and how they impact malting quality. And Christy's basically outcome of her PhDs was saying D-hordein is either the good guy or the bad guy, depending on your frame. So this is not new, but it was sort of putting it in context to do with starch quality at the time. But now we can sort of talk about the protein by itself. So we know that the storage proteins is around 50% of total protein. So if you have about 10% protein sample, around 5% is the endosperm storage proteins, or specifically the hordeins. But there's a range in that, and we'd seen ranges reported from sort of 40 to 60, 40 to 70. Well, we got to the samples that Marnie tested between 43 and 75% of protein content with the endosperm storage proteins, the hordeins, glutellins. So this is where you could have two samples, same variety grown at two locations, and it's both 10% protein, but the one protein sample, or the one sample that's 10% protein, it might only have 5% hordein and glutellin. And the other one might have 6% hordein glutellin. Do you think that would make a difference in any quality testing, any malting, any brewing processes? Maybe, maybe not. My gut tells me it will. The image on the right, I think, is one of the best images I've ever seen, and I will take no credit for this. I definitely did not take this image. But we see this sort of web-like structure here. Well, that's the protein matrix. And if we look towards the left, we can actually see, let me see if I can get a laser pointing. I'm pointing with my cursor here, and I know you guys aren't seeing anything. We see these lovely large starch granules here. And there's spaces where these granules were, but we see these tiny small granules, and they're completely embedded in the matrix. How difficult would it be for these granules to be released from the matrix? Maybe we need Kenny Reeves to help us release this matrix. The amount of work the enzymes have to do to break down this protein matrix around these granules is quite significant. And those enzymes are proteases, and we'll talk a bit about those. But in this matrix itself, it's not just the hordeins and glutellins. We know beta-amylase, the bound beta-amylase is stored in here as well. And the serpin-4, Z4 is bound in here as well. We also know bound up in here somewhere or somehow are these hordoindolines, these endosperm structural proteins that theoretically will connect the matrix proteins to the granules. And we know that a softer texture is actually better for malting because it allows the water to hydrate the grain more efficiently. So that's one of the things we talk about is improved hydration rates, hydration speeds as a potential benefit for the malting industry. You could assume that if you have less endosperm protein, then you would actually have more available access to the starch granules by the starch degrading enzyme. So look at this granule here. It's quite exposed, so I don't think the starch degrading enzymes would have any trouble attacking that. But some of these smaller granules, the starch degrading enzymes might have some difficulty in accessing those. And this applies across other grains as well, because you still have storage proteins in wheat and sorghum and corn and rice, etc., etc. But if we have a lower endosperm protein, then potentially we have lower amounts of beta-amylase. We can reduce the fan levels in wort because we've actually got less protein that's been hydrolyzed, and maybe reduce the foam positive. So when the industry says we need between 9 and about 12% protein, they've decided on that range for very specific reasons, or for very specific reasons, based on we need enzymes, we need fan, we need foam properties, but it's not too low and not too high. So that's the Goldilocks range for barley quality. And as I said, B-hordein has been shown to be the major limiting protein in advancing malt quality, and that was based on the work that Christy did way back in 1996. And this is one of the interesting things, and again, I will go on ad nauseum about the difference between the IOB extract method and the Congress method. The IOB extract method will allow you to see absolute differences in quality, whereas the Congress method may actually hide some of those differences because it's allowing enzymes to keep doing work. So the work that Marnie did, she basically got these samples, measured the alcohol-soluble protein content, and this is the relationship we saw between them. So we got samples from about 8% protein to some very, very high protein samples. And it does look like there's a general positive correlation between these. There's a general positive relationship between the total amount of protein and the total amount of the alcohol-soluble hordein fractions. That's good. But it's not a perfect relationship, and we do see this inherent variation scattering around that linear line. If you look at the samples just within the protein range, that scatter becomes even more pronounced. We see examples here where the total protein content was close to 10, but this sample had just over 4% hordein, and another sample about 9.5% protein, but it had 7% hordein. So my hypothetical question to everyone, do you think these two samples would malt the same? Do you think these two samples would have the same malt quality if malted in the same way? And this is some of the problems the maltsters face. They only know this information. They do not know this information, but they make some assumptions around what this could be. I think this is where we need to start looking closer at our grain quality. Not so much total protein content, but the storage protein content. And I know I do love to rattle cages and throw out really weird challenges to folks. So how does this relate to modification? And again, I'm focused on the malting side of the business here, not downstream in the brewing. We know that the storage proteins will be hydrolyzed by a huge number of proteases during malting. And these proteases is more complex than people think. And again, I've been around long enough to have seen work attempted in this space, both in Australia as well as the US and a little bit in Europe at different times, and trying to quantify and do specific assays for the different proteases. It impacts on the soluble total and the ratio of soluble to malt protein. It impacts on fan levels, which is something we're seeing and having conversations with brewers at the moment. And I did a recent podcast with the NBAA about fan and the risks of fan. Most brewers will think it's only the fan that's coming in from the malt, but they don't realize they're adding fan when they're adding hops. And in the US, as you know, they love to dry hop their beers. So every time they're dry hopping, they're adding a lot more fan. They just didn't know it. And we're actually testing beers where we're seeing more than 200 milligrams per litre of fan in finished beer, which may or may not be a problem. It just depends. We certainly know this can affect foam properties. If you push your modifications, then you start to lose some of those foam positive proteins. And we know we talk about beer haze. Well, for a very, very long time, we didn't want haze in beer. There was no way we wanted hazy beers. Now you can't turn a corner and go into a tap room and they don't have 2 or 3 hazies on their beer menu, and you think, wow, how the world has changed. So this is our group of enzymes activated during germination, specifically malting. It relates to their pH optima, the temperature optima, and when they start to denature. Very broad group. If we just look at the proteases, because we're only thinking about the protein story at the moment, these are the broad groups of proteases, sort of their pH, mostly the endopeptidase working in the mashing range, carboxylpeptidase also working in that range, that there are a couple of way outside the pH range of what we'd be working in malting and mashing. Again, temperature optima for these are quite low. So again, we lose activity in kiln, and we also lose a lot of activity in the mash. If we're mashing in hot at around 65 degrees C, well, most of these enzymes will be denatured fairly quickly, although it does suggest with those inactivation temperatures that if some of those are still active at around 70 degrees Celsius, well, then we will be seeing some protein hydrolysis towards the end of mashing, and certainly some increase in amino acid production towards the end of mashing. So this is all very interesting, and this is where some of the industry hasn't kept up with this knowledge, or don't pay enough attention to this knowledge that they could still be actually getting protein hydrolysis, even when they're mashing out at 72 or 74 degrees C. So just to sort of touch again on the proteases, it's very complex. Again, 4 classes of proteases. That is not based on their targets. The cysteine proteases do not target the cysteine bonds. It's really to do with the active sites. A couple of different proteases there in the cysteine protease family. And the EP1, or what is sometimes we call malt endoprotease 1, theoretically, that's the proteases that freeze up the bound beta-amylase in the endosperm. So again, there's been a lot of attention paid to it specifically. The serine proteases, and remember we have a serine protease inhibitor in the storage proteins, but chymotrypsin and trypsin. So these proteins are very common in nature as proteases, and I love to sort of stump my students by saying, what's the key protease in your stomach that's digesting protein? Well, it's this one here. Well, the pH of your stomach acid is about 2. So these proteins can function at a very low pH, but again, there's slight allelic variation in their genetic sequence to allow them to do that. These are the 2 proteases we use in proteomics. They digest nearly everything. We have an aspartate protease, which is pepsin. We see that in a lot of fruit, and some metalloproteases such as carboxypeptides, but we need some cofactors for these proteases to work well. And as we love to talk about, the answer to the universe, it is 42, if you know that joke. But coincidentally, to date, in barley, they have characterized 42 proteases. So when people say, let's develop an assay for proteases, yes, let's do that. Which protease? It gets complicated. So let's jump into the proteomics, and that might help us understand where we can actually really start to look at protease activity. This is part of Marnie's work, where she looked at about 8 different varieties of barley grown in the US. Some were varieties released from the University of California. So that's the UC. You see on the name there, University of California varieties. OSU is Oregon State University Full Pint. And there's a couple of introduced varieties from overseas as well. The Canadian Crop Center, Crop Development Center, Copeland is a common variety grown in the US as well. The locations, there were 8 locations across California and over 4 growing years. This wasn't a completely balanced set of trials because occasionally they would drop a variety out because it wasn't performing and they'd bring a new variety in, as you know. And a couple of the years, the trials got completely decimated with drought as well. So this is a heat map showing the either increasing abundance or decreasing abundance of very specific proteins. And Marnie actually identified, well, she got 3,000 hits for individual proteins. So that complicates the total protein content story a little, because we're now saying, oh, we got a protein content of 10%. Okay, what proteins are important? Well, we might have 3,000 proteins that could or could not be important. So generally, we'll look at the top 50 or 100 proteins. And while we don't have time to really go through this list in a lot of detail, we see one of our major softness proteins or textural proteins there. We see beta-amylase. It's in another softness protein there, the hordoindolines B. We start to see our hordeins themselves, hordein, D-hordein, gamma-hordein, B-hordein, et cetera, et cetera, et cetera. Go down the list. And we can see that in some cases, we see that everything's sort of a very high abundance. And in some cases, there's a mix across growing locations here where it's a low abundance versus a high abundance. So definitely the environment is influencing all of these protein contents. And in some cases, you might still have 2 samples from 2 locations. The protein content is still the same, but the overall variation composition is starting to really become the main story here. It's the main character of the play. If we just look at our main endosperm storage proteins, and yes, I did cherry pick these. That's my privilege to, or my right to cherry pick my data. No, we didn't do that. So this is an example of 6 locations grown across these 2 years. The total protein content, well, that basically tells you the total protein content. The TPC, the alcohol-soluble protein content, is the alcohol-soluble fraction, and the percent alcohol-soluble relative to total protein content. So in this example, this is all the genotypes at that location averaged. Yes, averaging data may or may not be the right thing to do, particularly with this sort of data. But we're seeing here between sort of 54% up to about 65%. So probably within that normal expected range. But we do see significant changes in abundance between some of these proteins, particularly between some of the individual subunits within a family here. It might be high at one location and it reverses. So it's just telling us that, yes, what's going on during grain filling is very important. The amount of available nitrogen might be influencing this, so the amount of fertilizer used, how that nitrogen is used during that grain filling period, what's going on during the weather in that grain filling period, how much is that influencing these abundances and the relative ratios to each other. Again, looking this in a little bit more detail, and that's Marnie's publication if you'd like to get that. It should be open access. Again, this University of California variety Capay, grown across these locations, across those years. Total protein content, we didn't have alcohol-soluble protein content across all years because basically in the early years we ran out of grain. They didn't harvest enough grain often for us to do some of this detailed analysis. But again, you can spend a little bit more time looking on that. But these are the ratios of these proteins shifting significantly. And just looking across here, the difference between sort of 8.9% protein and 9.4 is not significant, you would not think. And there's significant variation in the ratios as well as the abundance of these proteins. Now, these are the main endosperm storage proteins, and I would bet, what would I bet? I'd bet at least 1 pint of Guinness that this is having an impact on malting efficiency, these ratios and how much of these proteins are laid down. And that's just the protein by itself. So who's prepared to take me on with a pint of Guinness that these are definitely not or definitely having some effect? So the general protein relationships and a sort of that if-then scenario. The industry uses plump grain as a surrogate for saying if we've got nice big fat plump grain, well then we've got more starch. Perfectly true. If we have more starch, then theoretically we'll have more fermentable sugars. Absolutely. If we have more starch, then we'll have less protein. Probably. It doesn't always work like that. If we have less protein, then we probably have lower beta-glucan, which is good. It lowers the risk of runoff issues in the brew house, lower risk of filtration issues, possibly going into packaging, and lower risk of unwanted hazes. So here we have the story. Large fat grain, more starch, less protein. Generally, life should be easy. But what complicates this is we're talking about total protein content, not protein composition. And this is where it gets less clear. Now, do these major storage proteins have any impact on any starch properties? Well, they do. And here's an example from Wenwen's work where we see these negative relationships between the amount of these hordeins, and this was measured through proteomics, and the amount of fermentable sugar release using malt, and that malt was mashed in a hot mash, not a Congress mash. I refuse to use a Congress mash unless somebody says, please do a Congress mash. All my mashing will be a hot mash. So we see these negative relationships between the amount of hordein present and the amount of fermentable sugars. Something I didn't present was really how this all impacts the interaction with starch. So we have actually seen that the amount of total hordein, or the amount of alcohol-soluble protein, does actually impact gelatinization properties of starch. Because as I showed with that image that SEM image, those tiny small granules which impact gelatinization. The smaller granules will gelatinize a little later, or the temperature is a little warmer for gelatinization. If they're bound up with that matrix, they have less capacity to swell. And that's what gelatinization is about. This is granule swelling and bursting. So if they're bound up in that matrix, then they're physically limited in how they can swell and burst. So we know that higher protein content, you will end up usually with higher gelatinization properties of starch. But now we can see with higher alcohol-soluble protein fraction, we're definitely seeing some higher gelatinization temperatures. And if the growing environment is increasing the amount of alcohol-soluble protein, we could easily say that will impact gelatinization. So this is how storage protein is actually impacting mashing in the brew house. I got sidetracked, but you guys are used to that now. We see a range of the storage protein in our samples between 40 and 70% of total protein content. That's sort of not new. People have published that information before. But what we did, we developed an NIR calibration. So currently it's on ground grain, not whole grain, which is a limitation, I know. But it allows us to see the amount of alcohol-soluble or the endosperm storage protein, the alcohol-soluble storage protein in our samples. And as a consequence, we might be able to control malting a little more, knowing the amount of alcohol-soluble storage protein, and we might be able to adjust some steep times. This is pretty much controlled by environment. We did a G by E on the samples, which I'm not sure has been done before on proteomics data, maybe. Of all those major endosperm storage protein, the B hordein, B1 hordein was the only one that was influenced by genetics. All the rest were influenced by environment. Individual hordein themselves are influenced by environment. We know there's impacts on quality to do with modification rates, especially if you do the IOB mash, the hot mash compared to the Congress mash. We know that these will impact gelatinization temperatures and will also impact the amount of fan if you're using a hot mash. And as I said, we developed an NIR calibration, whether that could be used in industry to select loads and you could segregate on alcohol-soluble protein content rather than protein content. I know that's a huge ask in the industry. Yeah, I've worked with the industry for 30 years and I understand the whole logistics game and stockpile management. This is a whole new paradigm for them, but that's what they're looking for. And they could actually start designing their steeping schedules based on alcohol-soluble protein fractions. So in the relatively short time we've had together, hopefully I've presented you with some new information and maybe got a couple of you thinking about, is this something we need to explore a little more? Do we need to shift some of the focus to some of our more moderate climates? Is this the same we see in moderate climates versus our extreme climates, such as California? Always looking for innovation here, but we are sort of locked into this old paradigm, certainly here in the US, where they want to use the Congress mash rather than a hot mash. And I really don't think the Congress mash tells us enough about malt quality. Of course, we can sort of talk about these things and suggest changes for industry, but it comes down to what the farmers will want and how that would change what the farmers do. But I know the farmers are already concerned about productivity, planting times, inputs, all those sorts of things. So we would always have to have these conversations with them. Would we be looking at new protein segregations? Well, I don't know. That is a whole new conversation. We have a craft malting industry here in the US. There's around 130 craft maltsters, and they vary in size from 500 kilograms a week to 5,000 kilograms a week. They're still very small relative to the big bulk maltsters. This is possibly something they can get into because they could be segregating or accepting loads from farmers based on protein content, but also the alcohol-soluble protein fraction. And they could design their steeps accordingly. Now, some recent work published out of Australia, not with my group, but with Edith Cowan University in Western Australia with Michelle Colgrave's group. They've published a couple of works recently where they were showing that a set of lines they produced that had variation in the hordein families. So it was either missing one or more of the hordeins. And it did seem like the D-hordein was the most problematic. So if you could get rid of the D-hordein, things would generally be better. And I think this is the work that Stuart Swanson demonstrated way back about 20 years ago. The D-hordein was the more problematic of the hordeins. Can we grow barley successfully without D-hordein? Yes, we can. Can we produce malt and beer from barley that doesn't have the D-hordein? Yes, we can. Will it be beneficial in terms of overall improved quality, more efficient malting? I think it will, but Mother Nature has ways of throwing us a curveball. And if the amount of nitrogen to be laid down would be the same, then will another protein increase in abundance relative to that, and would that be problematic? So again, a lot of work to be done, and proteomics is probably the place or the technology where we can really start to explore that and really understand those relationships. So that's pretty much the presentation from me, and I'm more than happy to jump in on some questions.
Oh, thank you. That's a lot to take in. That was great. There's a number of questions already in the chat. If anyone would like to ask a question, either it's probably easier via the chat, I think, in this version of WebEx. The initial question came from Eskil Bergen. I don't know if she can, that can be asked online, or whether I have to voice the questions. I'll ask me. The first was any thoughts on the potential of blending grain batches at the far end of acceptable malting intake specification criteria to achieve a higher quality malt? Is there ways of separating out your batches?
Yeah, the industry has these processes, and a lot of maltsters will have this sort of as an in-house conversation and may not make it sort of publicly known. But they will malt a certain variety, and they might do 20 or 30 batches of that variety at different levels of modification. Then they might do a different variety, and they might then blend those two varieties at different ratios to meet some brewer's specifications. So that is a fairly normal process for maltsters to achieve what their brewing clients are asking for, because often a single variety cannot meet all the specs for a brewer. So they will blend, because you'll have some varieties might not be higher in DP, but higher in FAN, and other varieties might be higher in DP and higher in extract, so they can blend those different malts. They would never blend barley varieties before malting. They will malt the varieties separately, then blend the malts. So that's a fairly normal process for maltsters to do that, to meet their specifications. Unless, of course, a brewer is saying, I only want variety X, and then they will only give that brewer variety X. And that's a common process here in the US. You've got Anheuser-Busch and other big brewers say, we've got our varieties, you will only malt our variety and give us our variety. So maltsters will mix and match as accordingly. Yeah. And I think that's the challenge with climate change, is sometimes the batches they're getting, they're seeing these unknown, or they're finding out late, these unknown consequences on quality that aren't picked up in the standard COAs.
And actually leads on nicely to the next question from Benjamin Potts, saying, how do you think rising atmospheric CO2 levels might affect grain quality and composition?
There has been work looked at productivity in terms of grain yield and grain protein. And as my understanding, that's the limit of what's been done thus far. I'm going to be perfectly honest, the CO2 space, atmospheric CO2 space, is not something I've been paying a lot of attention to. I do get asked about CO2 in malt houses and the impact of not having enough CO2 or not getting rid of the CO2 fast enough in a malt house in terms of atmospheric CO2. It's certainly impacting productivity, and they're talking about increasing yields and those sorts of things and increasing protein content. The CO2 wouldn't be acting alone because there would still be the growing environment itself, the heat, the drought, the available water, nitrogen. So it would be a complex interaction, and how all of these things interact with rising CO2. I think it is very complicated, and sorry, Benjamin, I don't have an easy answer for you.
Okay. There's another question from Al here saying, how could the criteria for protein composition change for malting alcohol-free beer? Is there any trade-off for some of the components between processes, and would hot areas be better for such processes for alcohol-free production?
So yeah, some brewers might be looking at the malt quality and asking maltsters for slightly different malt quality for if they're producing a non-alcoholic beer, but it would depend on their process of producing a non-alcoholic beer. So are they mashing it at even a hotter temperature, say closer to 68 or 69 degrees Celsius, to reduce the starch degrading enzyme activity, which is there for potentially reducing the amount of fermentable sugars? Or are they making alcohol beer and just removing the alcohol? So there's different ways that they're producing these non-alcoholic beers. I think a lot of brewers will be just thinking about, this is my malt, and this is what I'm going to do with that malt. They're not actually sort of pushing that upstream to the maltster and saying, well, you need to now make me a malt that will make it easier for me to make a non-alcoholic beer. Errol, that's a really good question. I don't know yet of any breweries that are pushing this problem to the maltsters and saying, you make me a malt that will make it easier for me to make a non-alcoholic beer. But that is an interesting position to look at.
So is that a major driver in the industry, alcohol-free beers?
Oh, it's about a 30% growth in alcohol-free beer here in the US, and it's basically keeping our industry afloat. There's been significant decline in total beer consumption. The biggest hit has gone to sour beer production. People have moved away, moving more back to pilsners and lagers. We still have our IPA processes, but generally the non-alcs, all breweries now, if they're not making a non-alc, they are looking at how they can make a non-alc. It has really shifted the whole focus here.
Yeah, I think it's the same here. I think, yeah, something's happening. There was a question from Chris saying, how would you change the steep regime based on the alcohol-soluble protein ratio information that you have about the grain?
Well, there's two major limiting physical components within the grain when you're trying to steep. One is getting the water into the endosperm cells, so you've actually got to start to break down the cell wall. So there's the beta-glucan-arabinose island. But if you've got lower amount of endosperm storage protein, then theoretically you will have a softer kernel. So things will hydrate a little faster or a little more uniformly. So I think knowing that will allow you to potentially not necessarily change a steep protocol specifically, but maybe sort of give you that confidence you've got a more uniform hydration. Now I know some of the maltsters are looking to move to single steeps, not even having two steeps, having a single steep, and how long that steep, 10, 12, 14 hours, depends. So it might not actually change that, but it would probably give them the assurance that they'll have a more uniform hydration as part of that steep.
Okay, there's more questions coming in here. David Hall is saying this is fascinating. I was wondering about the picture of protein content variation across the field, one of the first slides. How was that made? Was there some kind of aerial measurement, or was that reconstructed from sampling positions?
David, great question. This was back in the good old days, when we would actually be working with the farmer, walking behind the harvester, grabbing samples out of the harvester, and taking a yield measurement, and then doing NIR on that sample. So I had a portable NIR carrying it around the field. So that's how we could sort of lay that yield map with the protein map. Now, modern harvesters, you can get on the go yield data, so real-time yield data, and you can get that with moistures, and some modern harvesters will give you protein data at the same time. So you can actually start to get more reliable yield and protein maps. And they're often using it just to see the uniformity of fertilizer across the field, like the nutrient range of their field. So do they need to add more nitrogen one end and less one end, so they can help them balance their nitrogen inputs or fertilizer inputs? So it is becoming more common. That was in 2006. So that was long before we had these technologies built on harvest. We were literally walking behind a harvester, capturing samples. We'd have the farmer stop about every 20 meters, which he ideally didn't want to do. But yeah, so there was nothing fancy then, but now there's a lot more common technologies on harvesters that'll do that for you.
That's a quick question from Sergio saying, are these alcohol-soluble protein testing equipment available to be used at an industrial level?
So we're just using NIR. It's a calibration we developed. We are talking with the company that's got us that NIR, whether we would make it a commercial product. The limitation would be, at the moment, it has to be ground. So for most grain handlers or maltsters, they really don't want to be grinding a sample. They want to put it through as a whole grain, just get protein content. So I think we're still a little ways from having it as a sort of inline measurement at grain receivable or malting. We're still probably at that validation. We've got proof of concept now. We're just got to move to that validation phase. But it's a standard NIR. There's nothing fancy about the NIR equipment at all.
Okay, we have a question from Raoul saying, increasing CO2 requires adding more nutrients to the plant to compensate for the excess carbon skeletons being fixed apparently. How can we ensure these blocks, nitrogen, phosphorus, etc., are not used to generate more proteins than sugars?
I'm not sure you can. The plant's got to do what the plant's got to do. It's got to survive. Its whole intention is to produce the next generation of seeds. And it will shift the balance of where it's moving its nutrients, whether it's going, if it's really lush conditions, then it knows it can produce more starch, and as a consequence will produce less nitrogen. It is really about what it needs to do to just produce enough seed that is viable with enough endosperm reserve that it could potentially germinate in the next season. I'm not sure. That would be an interesting concept, to actually try and block the plant from producing some proteins and producing more carbohydrate. That is an interesting concept. Again, Raoul, I'm not sure I've got a good answer for you.
And on a different topic from Eric, it says, in terms of protein quality, the focus is on beer. What about barley protein for human consumption?
That's a good question. I guess barley is seen as not the main grain in terms of human consumption when it comes to protein content or protein quality. Generically, we'll just go, we go to wheat. In terms of barley, in food consumption, we think more in the context of fiber, to do with beta-glucan or arabinoxylan. And there are definitely food-grade barleys out in the world that will be slightly higher in beta-glucan. And as a consequence, usually they're higher in protein anyway. I don't think the food industry would prioritize having higher protein barley as its main source of protein coming from cereals. I think they'd still stick with wheat, but then they're more than happily shift to pulses and legumes, which are in that 20% range of protein in terms of getting protein from grains and cereals. But again, the industry the whole barley industry is primarily focused on what the malting and brewing industries want. If the malting and brewing industries suddenly shift a quality target, then the breeding programs will shift to that. If the food industry says we want higher beta-glucan, not too many breeding programs are going to shift to higher beta-glucan because the majority of their barley is targeted at that malting and brewing industry. So I think the food industry is just going to be a take what it can get. It won't be in a position to dictate quality. I think it's just an unfortunate position the industry's in.
The way I've got my setup here, I can't see the participants if anyone raises their hands. Malcolm, can you tell me if there's anyone has raised a hand?
No, no one's got their hands raised just now.
Okay, well, maybe I can ask a question, given there's a little bit of time left. I was interested in the relationship you were finding between hordein levels and levels of fermentable sugars. Is that purely an aspect of this matrix, or are there other functional impacts in that relationship?
I think it's definitely a physical process where if you've got inherently more of these storage proteins, then potentially your modification levels might be slightly lower. As a consequence, then you may not have as much beta-amylase released. But also the limitation of these storage proteins impacting how quickly the small granules can hydrate in the mash. And if they're not hydrating, if the small granules aren't swelling and sort of erupting till they're getting to 70 degrees Celsius, then the starch degrading enzymes have lost a lot of their potential. So it is this sort of this physical barrier, but also this time-sensitive relationship between the substrate availability and the enzyme itself that's working on those substrates. And we're seeing, I mean, I'm seeing malt samples. I tested one for one of the bigger maltsters just before the new year, and we were getting offset temperatures in gelatinization around 71, 72 degrees Celsius. And they're concerned. And now maltsters are thinking they need to look at adding gelatinization onto their COA so they can give their brewers some guidance on mashing conditions, rather than brewers just saying, well, I'm mashing at 65. Okay, that's fine, but how long are you resting at 65? Maybe you need to go to 68 and 69 a little quicker than you thought, just so you can get that more of that starch solubilized.
Okay, thanks. And I do see a raised hand from Chris.
Yep, Chris, you can unmute yourself and ask your question.
Hi Glenn, thanks so much for your talk. It was really interesting. I had a question about whether there were any types of endosperm proteins that could facilitate water uptake, or do they all restrict water uptake?
Good question, Chris. Thank you. I think at the moment our focus has just been on the hordeins, and they're quite limiting in water uptake. We know that, and some of the works that they did at Edith Cowan in Australia, they saw that some of the nitrogen was being used then, if it's missing a hordein, a single hordein family or a combination of hordeins, then the plant is still using that nitrogen and it's making globulins. So endosperm storage globulins. So whether they would be a water limiting protein, I'm not sure. But in terms of just general hordein, they're quite hydrophilic. And that's why we see some of the short hordein peptides are part of the beer foam, because they're hydrophobic and they form foam, the bubbles in the foam. So whether some of those other proteins would be more hydrophilic, that's a really good question.
Thank you.
Are there any more questions? You've had a fair number there.
There's another question in the chat, I think.
Is there? Oh, thanks. Thanks, Malcolm. I'm glad someone's taking. Okay, yes. Yes, a question from Eric Stockinger. Beta hordeins are the only ones controlled by showing a genetic variation, environment affecting C and D. Does that mean there's no genetic variant for these gene families, or the environment overrides the genetic variation in terms of expression of C and D?
Yeah, thanks, Eric. Good question. So this was the set of samples that we tested, a set of varieties, and 4 or 5 of those were yeast, University of California, where they have a common set of parents. So there could be conserved genetics with that. On the flip side, if you've been around long enough, and some of us have, we used electrophoresis for variety identification. So you get a variety come in and they go, oh, well, we're not quite sure the racilla here looks okay, or maybe it doesn't quite look like that variety. Let's do electrophoresis. So there was enough uniqueness between varieties that we were using usually B and C results to say, yes, this is variety X. No, this is not variety X, it's variety Y. So there's enough distinction between the varieties there is enough variation. It just seems like the varieties we tested, there wasn't a big genetic component. But no, in terms of global barleys, yeah, there is big variation in all of these families, and it can be used for varietal identification. But generally, we would see environment driving the abundance. So if you did an electrophoresis, the intensity of some of these bands on the electrophoresis would change, knowing you've got a higher protein content because there's just so much of that protein being formed. You'd still see differences between them, but it's the intensity of some of these bands. So environment still definitely drives a significant portion of that.
Okay, I realize it's 5:15 here in the afternoon. Is there any other questions before we wrap up? If not, I could just ask everyone to show their appreciation and thanks. That was a great lecture. Learned a lot. And thanks again.
Thanks, everyone.
Keep an eye out for the information on the next IBH seminar in a few weeks' time. Okay, many thanks.
Thanks, everyone. Thanks, Luke. Thanks,
Okay,
Malcolm.
cheers.